The presence of these codons at the beginning of the frame give a clear indication of the translation initiation site. A always. B does not necessarily. C does not. D never. The correct answer is does not necessarily. There are possible stop codons, identification of which is straightforward. A5, B2, C10, D3. The correct answer is 3. Which of the following is a wrong statement regarding gene prediction using Markov models and hidden Markov models? A. Markov models and hums can be very helpful in providing finer statistical description of a gene. B. A Markov model describes the probability of the distribution of nucleotides in a DNA sequence. C. In a Markov model, the conditional probability of a particular sequence position depends on k alternate positions. D. A zero-order Markov model assumes each base occurs independently with a given probability. The correct answer is in a Markov model, the conditional probability of a particular sequence position depends on k alternate positions. Which of the following is a wrong statement? A. Prokaryotes include bacteria and archaea. B. Prokaryotes have relatively large genomes. C. Prokaryotes have relatively small genomes. D. In prokaryotes, the gene density in the genomes is high with more than 90 of a genome sequence containing coding sequence. The correct answer is prokaryotes have relatively large genomes. Which of the following is a wrong statement regarding TESTCODE method? A. This is based on the nucleotide composition of the third position of a codon. B. In practice, because genes can be in any of the 6 frames, the statistical patterns are computed for all possible frames. C. It is implemented in the commercial GCG package. D. It exploits the fact that the third codon nucleotides in a coding region fails to repeat themselves. The correct answer is it exploits the fact that the third codon nucleotides in a coding region fails to repeat themselves. Because a protein-encoding gene is composed of nucleotides in triplets as codons, more effective Markov models are built in sets of 3 nucleotides describing non-random distributions of trimers or hexamers and so on. A. True. B. False. C. D. The correct answer is true. In bacteria, the majority of genes have a start codon ATG or AUG in mRNA because prediction is done at the DNA level. T is used in place of U, which codes for methionine. A. True. B. False. C. D. The correct answer is true. Shine-Delgarno sequence, which is a stretch of purine-rich sequence complementary to 16S rRNA in the ribosome. A. True. B. False. C. D. The correct answer is true. The conventional determination of open reading methods identify only typical genes and tend to miss atypical genes in which the rule of codon bias is not strictly followed. A. True. B. False. C. D. The correct answer is true. The putative ORF can be translated into a protein sequence which is then used to search against a protein database. A. True. B. False. C. D. The correct answer is true. The use of Markov models in gene finding exploits the fact that oligonucleotide distributions in the coding regions are different from those for the non-coding regions. A. True. B. False. C. D. The correct answer is true. Which of the following is a wrong statement regarding the conventional determination of open reading frames? A. Without the use of specialized programs, prokaryotic gene identification can rely on manual determination of ORFs and major signals related to prokaryotic genes. B. Prokaryotic DNA is first subject to conceptual translation in all 6 possible frames, 2 frames forward and 4 frames reverse. C. A stop codon occurs in about every 20 codons by chance in a non-coding region. D. Prokaryotic DNA is first subject to conceptual translation in all 6 possible frames, 3 frames forward and 3 frames reverse. The correct answer is prokaryotic DNA is first subject to conceptual translation in all 6 possible frames, 2 frames forward and 4 frames reverse.